Okay, let's try this again. I'm trying to record this video for you for a long time. Remember that we'll probably, we'll either try to have a quiz next week on this and maybe some of the things that we've studied so far, or it will be after the winter break. But regardless, you do need to be studying what we've looked at so far. Okay, so we're looking at the plasma membrane, right? That's another word for the cell membrane. The different parts of the plasma membrane, and we're also looking at how things enter and leave the cell membrane. Okay, it is the gateway to the cell, how things enter and leave the cell. Okay, so here's an electron microscope picture of the cell membrane, and the most distinguishing feature that you can see here is that it's made up of two layers, a bilayer, bi meaning two, and two parts, two layers. It's a bilayer. Okay, it's very flexible and it can allow unicellular or one-celled organisms to move. Here on the side here we can see all these little cilia, and cilia are like little tiny hairs that can allow single-celled organisms to move. You have some cells in your body that are covered with cilia too. For example, cells in your respiratory tract, in your lungs, are covered with cilia that help move out pollutants from your lungs. That's why you cough, because those little hairs are moving, trying to clean things out. You also have cilia in your digestive tract that increase surface area that allow for better absorption of food as well. But for these little guys here, these cilia allow it to move. Okay, and the cilia, they are anchored right to the plasma membrane, cell membrane. Hey, again, here's this big word that we need to know. It comes up again and again and again. If you don't know it, please sit down and learn it. You need to know these words. Okay, homeostasis. Homeostasis. You need to know what it means. You need to be able to read it. You need to be able to write it. Homeo means same, and stasis means to stay or keep the same. Okay? So we looked at this before in terms of an important characteristic of life. All living things maintain homeostasis, which means they maintain a constant internal environment. So inside your body, right, for example, your body temperature is the same, right? Blood pH tries to be the same. Water content tries to be the same inside at all times, right? If these things, if homeostasis is not maintained, that's an indicator that you're very sick. Your body can't take conditions that change rapidly. So just like your body can't inside your cells, it's the same way. Okay, you can think of it as kind of an equilibrium. It's like a balance. Okay, for your cells, this is maintained by the plasma membrane. How? By controlling what enters and what leaves the cell. Okay, the functions of the plasma membrane. Okay, it serves as, number one, it's a protective barrier. It is known as being, it's a key word as well that we should know. It's selectively permeable. Permeable means the things that can kind of how it enters or leaves the cell, and selectively means it can select what it wants to enter or leave the cell. Okay, it's a bit choosy. Okay, it performs cell recognition functions. That's important for your immune system to recognize what's a foreign kind of bacteria in your body versus what's an actual cell that should be inside your body. Okay. We looked at just like your body has structure right from your skeleton, your individual cells have a cytoskeleton as well. They're made up of these long kind of filaments, and they provide anchoring sites for the cytoskeleton. Okay, enzymes. At this point, we should really know what an enzyme is, right? An enzyme is a specialized protein that carries out a chemical reaction, right? They provide sites for these enzymes to bind, okay? Cells are also together, they can adhere, they kind of stick to each other, and these provide sites for cells and tissues to kind of come together, right? And of course, the plasma membrane kind of encloses all the cytoplasm or the fluid in the cell. All right, so let's look at the different parts of the cell membrane. Okay, the most important feature of the cell membrane, which you need to know, is it's made up of a phospholipid bilayer. Phospholipid bilayer. Okay? These individual guys with the red kind of heads and two long white tails, these are called phospholipids. Many of them come together in two layers to make a phospholipid bilayer. Phospholipid bilayer. You need to know this vocabulary to be able to read it, write it, recognize, and talk about it in sentences. The other feature is you can see embedded inside the phospholipid bilayer are cholesterol. Cholesterol. Okay, we'll talk about what the function of that is later. Also, you have proteins that are embedded inside the membrane as well. You have two kinds of proteins. Number one, you have an integral protein. Integral. Okay. A key is inside the word. It's an in the word integral, meaning it's inside or embedded right in the phospholipid bilayer. It goes all the way through. Okay, that's different from a peripheral protein. Peripheral, right? Periphery means on the outside. So it's kind of on the outside. It doesn't go all the way through the phospholipid bilayer. And we can see that this peripheral protein is a binding site or an attachment site for the filaments that hold together the cytoskeleton. Okay. In addition to these kind of proteins, the integral proteins and the peripheral proteins, there are proteins called glycoproteins. Glycoproteins. Glyco meaning sugar, and of course, sugar bonded to a protein. Okay? These sugar proteins make up something called the glycocalyx. 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 I hope you are repeating along with me while I'm saying these. Glycocalyx. Okay? Let's go through the different roles for these, okay? Phospholipids, they provide the main structure of the plasma membrane, right? We'll talk about it, but they allow water to be attracted to either side, but not water to pass through. Okay? Cholesterol kind of maintains the spacing of the phospholipids, right? If it gets too hot, right, they want to kind of expand and go away from each other. So the cholesterol kind of keeps it together, right? If it gets cold, they want to pack very closely together. So it maintains distance, a good distance between the phospholipids, right? It's hard to see here, but this stuff is fluid like water. It's kind of sloshing and moving and flowing back and forth. So a good distance needs to be maintained between the phospholipids, but the distance can't get so great that it kind of rips apart. Okay, we know proteins, they're involved in many things. So these provide channels for things to move through. They provide anchoring sites for the cytoskeleton. They are involved in chemical reactions in the case of enzymes. Okay, the glycocalyx, right, provides kind of a fuzzy sort of layer on top of the cell membrane. One of the most important things it does is it's involved in cell recognition. So your immune system knows which cells are yours and which cells are not yours. Okay? They're also involved in cell communication so that different cells in similar tissues can talk to each other. Glycocalyx. Okay, we talked about that. I'll provide just the PowerPoint. If you want to read through it on your own, that's fine too. But we talked about these things, right? Cholesterol changes how fluid the membrane is. And the great thing about it is it allows the membrane to resist changes in temperature. The glycocalyx is very complicated, allows for a lot of things. You should know a few functions of the glycocalyx. Okay? All right. So what are the structure of the phospholipids, right? The phospholipids are the main structure that make up the plasma membrane. Remember, they're organized into two layers, okay? These are the main components that make up the cell membrane. Okay. On the bottom are two fatty acid chains. They're a lipid. We looked at these unit 1. The different macromolecules that we looked at, proteins and lipids, carbohydrates, nucleic acids, the 4 main macromolecules that make up life. So we've got 2 fatty acid chains, right? And of course, this is a lipid, so it's also attached to a glycerol molecule as well. The thing about fatty acids, right? A fat, right? You've probably done this as a kid, right? Tried to mix oil and water. The thing is, oil is nonpolar, which means that water, which is a polar molecule, will not dissolve in oil. It will form a layer. Right? You've tried this before, and that's what it means. So this part in the bottom really doesn't like water. Okay? The top has a phosphate group, which is PHO4. You don't really need to know the structure of this. This isn't important. If you take AP biology, the next biology course, that will be important, and you'll need to know more precisely how the chemistry of the molecule works. But for now, you need to know that it's got a phosphate head in which it's got a bit of a charge to it, which gives it a polar character, which means since it's charged and it's polar, water will like it. Okay? Okay, we describe the plasma membrane in terms of what's known as the fluid mosaic model. It's fluid because it's all sloshing and moving and flowing just like water. So like a liquid, okay? So it's a mosaic because the word mosaic, like in art, right? We've got a piece of art that's made up by lots of little pieces. The plasma membrane is made up of many different parts. And that's where the word mosaic comes into. Okay, you've got proteins, you've got cholesterol, you've got phospholipids, you've got glycoproteins, you've got all these different things that make it up. So it's like a mosaic. Okay, so back to our little guys, the phospholipids, right? Which this is a kind of drawing that's very simple, simplistic. This is the kind of drawing that you might need to do on a fun test. Okay, it's got this little head on it, right? It's charged, it's polar, so it likes polar molecules, right? We say that the head part is hydrophilic. 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 Another key vocabulary word that you need to know. Okay, a big hint that you should know this word is that it's in yellow. The color is different. The word hydro means water, and philic means to love or to really like it. Okay? This is the same feel. It's the same sort of root word that you find in philosophy, right? Philosophy is the love of kind of Sophia, right? The love of wisdom, right? In this case, it's water loving, which means it attracts water to it. Okay? The tail parts that are made up of those fatty acids, right, that are non-polar, right? You've seen this as a kid when you tried to mix oil with water. They don't mix. They are hydrophobic. Hydrophobic. They're water-fearing or water-hating. Okay? You've probably heard of the word phobia before. He has a phobia. They don't like heights. They're acrophobic, or they don't like crowds. They're agoraphobic. They don't like spiders. They're afraid of spiders. They're arachnophobic. Okay? Or if they don't like clowns, they're chlorophobic. Okay? There's many different words. Okay? There's some bizarre ones that I can't think of right now. We're afraid of the number 13. Triskophobia or something like that. I can't quite remember. It's a weird Greek word. Okay? The point about these phospholipids, right, with the polar hydrophilic head and the non-polar hydrophobic tail, is that they make the membrane selectively permeable about what can pass through, right? Obviously, water's not going to be able to pass through that easily. It's going to need some help. But other chemicals can. Ones that are lipid soluble, right? Meaning if they can dissolve in a fat, they're going to be able to pass through very easily. Okay, so something like oxygen can pass through very easily, right? Which is what we need. We need oxygen to be able to carry out cell metabolism. Oh, there's that other big word, metabolism, using energy that we need to know. All living things, a characteristic of life is metabolism, right? Being able to use energy. Okay, carbon dioxide, which is always for animals, right, is a waste product of metabolism. It needs to get out of the cell, right? The reverse is true for plants, right? Okay, here's what it looks like. Okay, the word aqueous means water. Again, Again, we need to know these kind of root words. Hydro is a word for water, and the word aqua is a word for water, right? Aqua man, right? He lives in water. He likes water, okay? An aqueous environment means a watery environment, okay? So we see the polar heads are in, right here in the middle, this is our cell membrane, right? Made up of our bilayer of phospholipids. And we see the phosphate hydrophilic heads, both in watery environments, aqueous environments, and we see the fatty acid chains are in the non-hydrophobic area. They make up kind of the middle of the plasma membrane. Okay, again, the cell membrane is made up of how many layers? 2 layers. What do we call it? A bilayer. Bi means 2, right? A bicycle has 2 wheels, right? A tricycle has 3 wheels, okay? It's a bilayer of phospholipids, okay? If you start learning these root words, which I try to push them, they will help you learn and figure out the meaning of so many other words in science or your other classes. These words form the basis of so much academic vocabulary that you really need to know. Okay? 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 So solubility. Can things dissolve, right? So we look at, you put salt, and you mix it in with water, right? The salt doesn't stay in with the water, right? It dissolves in the water, and you get this kind of salty water, right? It's great for making soups taste better, okay? But oil, right? Some things can dissolve in oil as well. So things that are non-polar, we always say with solubility, things that are similar, that like dissolves like, okay? So materials that are soluble in fats and lipids, right, can pass through the cell membrane easily, okay? Things that are not won't, okay? Of course, this makes, again, what's important, the semi, which means a little bit permeable, able to pass through membrane. Okay, so we've got small molecules and larger hydrophobic molecules can pass through the membrane easily. And we're looking at things, for example, oxygen, carbon dioxide, and water. Water is a bit of a special case that needs a little bit of help, and we'll look at that. Okay. If we've got ions or hydrophilic molecules and ones that are larger than water, or things such as proteins, they can't pass through the membrane easily, and they're going to need some help. So we'll look at the ways that they can pass through. Okay. So which leads us to our next topic that we've been talking about, how things get across cell membranes. Okay. The easiest way is through the process of diffusion, right? Diffusion is just, it requires no energy, and it just is when molecules move from areas of high concentration, where there's lots of them here, right? This is an area of high concentration. The little dots represent molecules or particles, and they're going to spread out and move to areas of low concentration, where there aren't any there. Okay? This is why you spray an air freshener in your dorm room, right? You can originally smell the air freshener near you where you sprayed it, but it doesn't just stay near you, it spreads out all across the room, right? And your roommate on the other side of the room in about a minute can smell the air freshener too, because it's spread out. It's moved from an area of high concentration to an area of low concentration. It's diffusion. Okay? We say that diffusion is a passive process. It's passive rather than active. Okay? Passive means that it requires no energy. Okay? Why? Because we know, remember way back we talked about particle theory, and one thing about particles, molecules, and atoms is that they're always moving. They're always moving unless you can get the temperature down so low that they stop, and that's called absolute zero. But just for our purposes, they're always moving, so that means that they have a certain kind of kinetic energy. Kinetic energy is the energy of movement, so they're just going to naturally move out. Okay? And so here's our picture of the cell. We've got a very high concentration outside of the cell and a very low concentration inside the cell. So these particles are naturally going to want to move in and establish a kind of equilibrium. Okay? So we can see this with food coloring, right? We put a little bit of dye in the water, and it starts off as a very concentrated blob in the water. And then it starts moving out from its high concentration to its area of low concentration. And eventually, if you wait long enough, the whole glass of water is evenly colored, right? It's maintained a state of equilibrium. Okay? And that's what happens. Here's an example of this plasma membrane. It starts with an area of high concentration. There are many of them there. And inside the cell, there's low concentration. So they're going to eventually move across, right, until you get a sense of equilibrium. So we say that things move with a concentration gradient, or they move down a concentration gradient, from high to low, right? Okay, so a special kind of diffusion for water is what we call osmosis. Again, this is another big vocabulary word that we need to memorize, to learn. Osmosis is the diffusion of water across a membrane. Okay? And so we have a semi-permeable membrane. Okay, we're going to move. Water will move from high water potential in areas of high water concentration, which means that water usually has things dissolved in it. Those things that are dissolved in water, such as salt, is known as a solute. Okay, the thing that does the dissolving is a solvent. The thing that's dissolved in it is a solute. Okay, so it goes from high water concentration or low solute concentration to low to high solute concentration, okay? Okay, this will be clear. The thing is just to remember that the water part, sometimes you get confused about the solute part in there. Just remember it's going to move from high water concentration to low water concentration, okay? Or it's going to want to try and dissolve more of that solute that's in there, okay? So here we've got an example of, so here, look, we've got our phospholipid bilayer, okay? These little blue guys here, these are water molecules, okay? And the green dots are the solute. So they could be an example of, like our experiment the other day, they could be an example of salt, sugar, or starch, okay? So as we can see here, we've got high water concentration on this side, and on this side, the right-hand side, we've got very low water concentration, okay? On this side of the membrane, we've got a very low solute concentration, green dots. On this side, we've got a very high solute concentration. So we're talking about osmosis, which is the diffusion of water. So the question you always need to ask yourself is, which way is the water going to move? Is it going to move into the cell or out of the cell, or is it going to stay the same? In this case, so yeah, it moves across. Sometimes you get confused about, oh, this is going from low to high, but you said it goes from high to low. Yes, so we're talking about water. So water is going to always move from a high water concentration to a low water concentration, right? It's going to try to dilute this extra solute. So when we're talking about osmosis, just sometimes you get confused about the solute and you get these kind of percentages, 10% salt. Just think about which one has more water. It's going to move from the one with more water to one with less water. Okay. Now, some of you that are very sharp should be saying, but Mr. Harrison, you said in the middle, these fatty acid chains are very hydrophobic, means they hate water. So how can water pass through there? Well, that's right. We need these special proteins that are used that make a kind of channel that allow water to pass through easily. And these are called aqua. Again, there's that word aqua, which means water, and porin. A pore is like a little hole. So it's a little hole or channel for water to move through, these aquaporins. And they're protein pores that are used during osmosis, okay? Because we've got the protein here on either side, it kind of blocks out the effect of these hydrophobic fatty acid tails. Okay, so there's three kind of solutions that we'll look at, and then this will be the end of this video because this is how far we are in class, okay? So the first one, so this yellow thing here is a cell, and this is kind of the aqueous or water environment that's outside of the cell. Okay, so we're just looking at one cell in a solution. Okay, so this is what's called an isotonic solution. Isotonic. Okay, the word iso in Greek means the same. Okay, we've got the word homo or homeo that means the same. We've also got the word iso that means the same as well. So what this means is that it's going to be the same kind of solution inside the cell as outside the cell. Remember, we're talking about osmosis here, so we're talking about the movement of water either inside the cell or outside the cell. So, the big thing to pay attention to is the water concentration. And if we look at the water concentration on both sides, it's the same. Both of them are 90%, right? So water moves from high concentration to low concentration. Since the concentration is the same on both sides, there's no water moving in an isotonic solution. It's perfectly happy. So what's the direction? There's no movement of water. Okay? The cell is at equilibrium. It's equal on both sides. Okay? Now this is called a hypotonic solution. Hypotonic. Now it's hypo not in terms of water, but in terms of solute. So we put the cell, it's outside of the cell, the aqueous environment outside of the cell is in a hypotonic solution, which means that it has less dissolved solute, NaCl, sodium chloride, which is salt, right? There's less, there's 10% outside, there's 20% inside. So this is in terms of the solute concentration. But in terms of where the water is going to move, right, it always moves. Osmosis is diffusion from high concentration to low concentration. So outside of the cell, there's a lower solute concentration, which means inside the cell, there's a higher solute concentration. But if there's a higher solute concentration, there's a lower amount of water, right? So what this means is in a hypotonic solution, the water is going to move from the outside into the cell, right? It goes from high water to low water. Okay, it's going to move inside the cell. And what can happen in this case is that the cell will get larger. It will swell, right? Think about our experiment with the dialysis tube. We're expecting to see an increase in mass. Where does that mass come from? From water that's going inside the cell. Okay? Now, the third kind is called a hypertonic solution. Hyper means more here. More solute, not more water. This is the only tricky part about it. The movement of water is just from high water concentration to low water concentration, but in terms of the solution we're talking about means that it's got more dissolved solute in it. So this one, the aqueous environment on the outside has a 15% sodium chloride or salt solution, which means it's got an 85% water on the outside. The inside, right, it's got very little solute, but it's got more water. So the water for osmosis always moves from high concentration to low concentration of water. So it's going to move from the cell to the outside of the cell. So if it's going to lose water, we would say that it's going to decrease in mass, the hypertonic solution, and it should kind of shrink or shrivel up. These are questions that could appear on a test. These are classic test questions. Mr. Harrison does an experiment and places potatoes in 3 different solutions. What kind of results would you expect to see? This kind of stuff. The movement is out. Okay? So this is a classic kind of experiment that's done with red blood cells, hematocytes, red blood cells. And then, you put it in a hypotonic solution, which means that there is more water on the outside than the inside. Remember, hypotonic means the amount of solute, which means there's less solute on the outside, which means there's more water on the outside. So it's going to go inside, and we can see the cells expanding. We've got the hypertonic one. Ooh, the cell is deflating or shrinking, right? Right, which hypertonic, right? Which means there's more solute, which means there's more water on the outside. And isotonic means it's the same, so there's no difference there. Isotonic solution, there's no net movement, things are fine. Our red blood cell here, it's going to be okay. Hypotonic solution, what can happen is cytolysis, or cytolysis. Anyways, the word cyto means cell, and the word lysis means to cut. So what we get here is the cell membrane. It breaks or splits open, and the cell can die as a result of being in the hypotonic solution, right? Because there's so much water that's coming in. We got to think that there's now a lot of pressure. We call that special pressure inside the cell due to water in the cytoplasm. That's called turgor pressure. Turgor pressure. And it pushes against the membrane so much that it can break, right? You think about a balloon, right? You can only put so much air in a balloon. You only get so much air in there before it pops. And this is something very similar to what's happening with the cell. In a hypertonic solution, it's losing, and what you can get is plasmolysis, right? The cutting of the membrane and going out, right? Ooh, it's all going to leak and shrivel up. Okay? So with cytolysis, right? Ooh, the cells swell and they burst, they get big and pop your balloon. And for this one, plasmolysis, they shrink and shrivel up. Okay? And this is what happens in your red blood cells. And here's 3 examples. This is a normal red blood cell, and it's got the nice red color. And the thing is, the middle of a red blood cell has got this little bit of a dent in it, right? One of the key features of red blood cell is that they are denucleated cells, which means that they lose their nucleus after they first develop. And the reason why they do that is so that they can hold more oxygen, right? The function of a red blood cell is to carry or transport oxygen around your body, okay? This one here, so this is in an isotonic solution. This is a normal looking red blood cell. This red blood cell in the middle, if we look at it, we can't even see this little dent in the middle anymore. And why? Because it's gotten so big, okay? So this is your hypotonic. And this one's kind of starting to shrivel up, and it's got a weird kind of shape that you can see. And this is your hypotonic solution, isotonic. Okay? Okay. And a term that can refer to these different kind of solutions is tonicity. Okay, so you should be able to, if you got a diagram like this, to look at it and decide, which one is isotonic, which one is hypertonic, which one is hypotonic. I'll give you a second to think about that, or you can just kind of pause it. I'll give you a minute. These are red blood cells, and if you think about it, which one is the normal red blood cell? Well, the normal red blood cell is the one with kind of the dented center in it. Okay, this one has this little line that's here trying to show you the dented center in it. So this is a normal-looking red blood cell, which means it's not bigger, it's not smaller, so it must be in the isotonic or same solutions. This one's isotonic. Okay. This one that looks like a big ball, it's obviously swollen up from this one. So this one's in the hypotonic solution, right? Because that means that inside the cell, right, there's less water and more solute. So water is going to go inside. This one's the hypertonic solution, right, which means that there's more solute outside, which means less water, so the water is going to leave the cell and go outside. Okay. Can we guess? So these are plant cells at the bottom. Remember, the thing with plant cells is they've got that cell wall around them, right? Plant cells really want to maintain, to keep their water. So one way they can do it is with the cell wall that provides a little bit of extra protection from water loss. But if we look at it, which one looks like a normal cell, which one looks like kind of a stressed out plant cell? So in the middle here, we've got our isotonic one. It looks normal. This one looks a bit expanded, so this one will be our hypotonic. This one looks a bit stressed out. It will be our hyper. Let's see if we're right. Hypo, yep. Hyper, yep. Isotonic, yep. Hyper is kind of shriveling up a bit. Iso, hypo. Good. Okay. And then we're going to talk about the different kinds of transport across the membrane next. So, what did we look at? The different parts that make up the plasma membrane. And we looked at simple diffusion across, right, which is a passive form of transport that doesn't require any energy. And we also looked at osmosis, right, which is the diffusion of water across the membrane through structures known as aquaporins. And how water gets across the membrane will depend on the tonicity of the solution, right? Whether it's an isotonic solution, a hypotonic solution, or a hypertonic solution. Okay, again, please, one of the main things you need to do for studying is to make sure all those important words you know. Please be practicing. Okay, which may mean that you need to slow down the video in some parts and watch it over again. Think, oh, what did Mr. Harrison say? I don't get it. I need to listen to this part again. It probably also means you can't just be passively sitting there listening. You should be taking notes as well to get your full brain engaged in what you're doing. Okay, good luck.